Well, thank you for the introduction and fantastic, Lara, for that chat. That was really cool. I actually work in 2D materials now, so we can have a chat after this if you'd like. Love to pick your brain. Hello, everyone. My name is Justin. Today I'll be discussing the development of smart sensors designed to mimic human senses, particularly the tongue. But before we begin, I'm sure you're wondering who I am, so I decided to give myself a little brief intro. As you probably guessed by my accent, I'm from the US. I did my undergraduate in biomedical engineering at the University of Texas at Austin and worked in Austin for a number of years. Then I moved to the UK to pursue a PhD in biomedical engineering at the University of Glasgow, where I focused on developing devices capable of manipulating light for chemical and biological detection and full color displays. And now I live in Barcelona working for the Catalan Institute of Nanoscience and Nanotechnology as a senior postdoctoral researcher. I work on the team to design, fabricate, and test advanced electronic medical devices to interface with the brain. But enough about me. Let's get back to the talk. So why would we want to develop devices that mimic human senses? And the answer is quite simple, actually. Nature does it best. Artificial devices that mimic senses are actually all around you. The microphone that you hear me speaking on, the camera that you see my beautiful face are examples. So are pressure and temperature sensors, and those can all be related to hearing, vision, touch, etc. But what about taste and smell? Well, for these two senses, there's an extreme challenge due to the complexity of how they work. Both smell and taste are used to help identify complex mixtures and often work in tandem to do so. So first, let's start with defining what is a complex mixture. Complex mixtures are the combination of many components in inexact proportions. So examples would be tap water, soil, your breath, beer, coffee. All of these are like a mixed bag of candy. So the coffee you drink, for example, may taste the same day to day, but the exact proportions of all the chemicals inside your cup actually do vary from brew to brew, and even with time as the coffee sits on your counter. Scientists typically identify complex liquid and gas mixtures using the gold standard method called chromatography, which involves the isolation of individual components of the mixture and identification of the percentage makeup of each component of the mixture. So to achieve this, we use bulky, complex, and costly equipment that is a very time-consuming process and can be wasteful. But when it comes to your tap water, you're not exactly interested in the breakdown of all the chemical components or what's in it. You mostly just want to know, is it safe to drink? Does it have a metallic taste, right? In the cases where identification of all of the components of a mixture is not necessary for identifying what that mixture is or determining if it's safe, a different approach can be taken than the standard methodology of breaking it down via so we use the sense of animal taste and smell as our guiding principle. So how do the senses of taste and smell actually work? So let's use chocolate as our example of a complex mixture. Everybody likes chocolate. So say I were to give you a piece of chocolate, but not tell you what it was. So while eating this unknown substance, the olfactory receptors in your nose and the taste buds in your mouth will each provide a unique response to the chemical mixture of chocolate. We call the response of your taste buds and olfactory units partially selective because each element to a varying degree interacts with all of the components of the mixture in your tongue. So in your tongue, for example, there are taste buds that have varying levels of sensitivity to a particular flavor. And each of these varying sensitivities have varying intensities. So you get a very, very complex behavior out of just taste in itself. So as all of the signals from the olfactory units and the taste buds go to the brain, this is where the magic happens. Your brain takes all those partially selective responses and tries to identify what it is you're tasting. So if you've tasted it before, you immediately recognize it as 85% dark chocolate, and that's that. But if not, your brain tries to decide if it tastes like something you've had before or not. So you may be able to identify it as dark chocolate, but maybe not the exact percent, or maybe you just identify it as chocolate or candy or just something sweet. Or maybe you're not able to identify it at all. We call this process pattern-based recognition. Your brain looks at the amalgamation of signals from the complex mixture of chocolate, and it receives from the nose and the tongue all these different variables, and it tries to identify a pattern in all of these signals based on your previous knowledge and training and experience to identify what it is you're actually sensing. So the accuracy and ability to identify is based on your prior knowledge. If you've never had this particular type of chocolate before, you'll have a very hard time accurately identifying it. Additionally, it is important to note that identification of chocolate is achieved without needing to know the identification of all of the individual components and chemicals, like sugar, that make up the chocolate, nor the exact percent of the composition of each component in that mixture. So looping back, you can already begin to see where chromatography, which works on the exact breakdown of all the components and the percent makeup in the mixture, may not be necessary. Turning to the model of the animal senses of taste and smell would therefore be a very, very good method to try. So rather than isolating the individual components of the mixture, like in chromatography, using this method, we create a pattern-based recognition map based on designing multiple different sensors that cross-react over a variety of sensors. So to do this, we make a bunch of regions kind of like taste buds of your tongue, where each region contributes its own unique signal for a given mixture. And by analyzing the group of sensors as a whole, we can then train our device to be able to discriminate, identify, and classify known mixtures. So the sensing platform we use is based on the phenomena known as plasmonics. So this slide is the most physics-based portion of the talk. For those of you who immediately glazed over on the mentioning of the word physics, I promise it is just one slide, so I'll try to get through it quickly. So plasmonics in a nutshell: when light interacts with metal structures in the nanoscale, a unique set of optical properties emerge. And the specific response elicited is highly tunable. The size, the shape, the material of the nanostructure, and the local environment surrounding it play a major factor. This response is also highly sensitive to minute changes of any of these parameters. So as the interaction involves structures on the nanoscale, this should allow for highly miniaturized devices capable of ultra-high resolution. So how did we create our artificial tongue? So to create our plasmonic-based tongue, and we made a bunch of taste buds. Each of these taste buds were a different region of nanostructures. And we modified each region to have a different response to the same mixture. So each different taste bud was modified with a molecule attached to the surface of the structure. Doing so altered the way the local environment and the structures within a region interact. So this effectively altered the affinity of each of the components in the complex mixture in a solution to our sensing device. So from that boring physics slide a minute ago, basically I've changed the local environment around the structure, therefore I've changed its response. So in the past, people have done this using multiple arrays of single metal nanostructures. But for our metallic taste buds, we actually effectively combined two different metals in one place, which had not been done before. So we combined gold and aluminum to create multiple sensing regions that we modified with different surface chemistries, as you can see by the bimetallic tongue image here. So in other words, a detection in one region gave us a unique response from the two different metals independently. And what's really, really cool about using these two metals, gold and aluminum, is that they actually have different chemistries that affect them. So we can ultimately alter just the aluminum or just the gold independently of one another without affecting the other. So it allowed us to create a really robust sensing platform. So our device looks something like this. For a size comparison, we have a 1 pound coin on the left-hand side. And each little colored square that you see is a tiny taste bud composed of millions of metal nanostructures. So this red boxed region here shows you one tongue, and there are 5 on this chip. It's worthwhile to note that this tongue is this size because we wanted to make it able to be seen with the eye. But in theory, we could have made it much, much smaller since each unit of nanostructures is 300 nanometers. So to test the capabilities of our tongue, we figured we might as well use the most common complex liquid we can find in Scotland, whiskey. Oh, and we also use whiskey to test our artificial device too. So we collected the response from each of our little tiny metallic taste buds and put them into a giant matrix. Then we used statistical analysis techniques to effectively obtain a pattern map and train our tongue. And this is what that resulting map looks like, right? So you'll see there's a bunch of different colors, and each color actually corresponds to a grouping based on one unique complex mixture. The closeness of each cluster to each other shows how similar each of those complex mixtures are to one another. And using the statistical technique called leave one out cross-validation, we actually found that our tongue was 99.7% accurate in identifying between these whiskeys plus the vodka and ethanol solution. So here's a 3D rendering showing further separation of the three Glenfiddichs tested, taking it beyond just a 2D plot, but a 3D plot using these statistical analyses components. And you can see as you add the third dimension, they are further separated from each other in space. So these results were actually published in nanoscale last year, and they got quite a bit of media attention. We were even on an Amazon Prime show trying to test our tongue versus experts who are whiskey lovers. So in summary, electronic tongues are based on our sense of taste, and they're made up of multiple cross-reacting sensors that need training and to be able to identify and classify mixtures. They are most useful in identifying complex mixtures from a known sample set and can be used to identify when something falls out of bounds of that set. For example, if tap water from a well source is no longer in the safe zone for consumption, or a whiskey is counterfeit, or if a beverage off a factory line still is correct in terms of its taste by spot checking. For a known sample set, electronic tongues are also capable of high accuracy of identification. And they can be made in a highly miniaturized manner, making them portable, and they also require only small sample sizes for operation. They can also be designed for particular application in mind. So for example, we can highly modify our device so that it is specific for testing a particular set of known structures where we can alter the metal or the molecule attached or the size and the shape. And this allows us to have a very robust system. And so yeah, and that's my talk. You can find me on Twitter, by the way, at Jusper. And these are all the people I'd like to thank. So I will now open up to questions.
Thank you, Justin. So we've got a question from Dory. Dory, do you want to unmute yourself and come and ask it?
Hello.
Hello, how are you?
I'm good, thank you. Thanks for your talk. That was really interesting. I was just wondering whether your tongue could also work for fumes and so like can work as a nose.
Yes, so the same exact techniques can be used for a nose. The problem is that gases are a little more complex to contain in a fluidic system in a lab. So we could have done that, we just did not.
Okay, cool, thanks.
Yeah, no problem.
I see that Matt's also got a question.
He doesn't have a mic. So
asks,
what Matt asked, go ahead, Bryn.
Is each metal tongue reusable, or can it only be used once?
So the really cool thing about using plasmonic devices for sensing mechanisms is that they are not consumed by the process. So you can make tongues and noses out of polymers as well, but those typically get consumed by the process and then you have to make a whole new set. For these structures and this sort of a design, the idea is you build a flow chamber and you can flow over your device a sample that you're trying to test for, and therefore it's reusable. No problem.
I also have a question.
Yes.
So we saw that the tongue itself was small compared to a pound coin. How is the device read? Do you like slot it into a machine? Have I missed this somewhere on the way?
So this device actually works by shining. So we transmit light through it. Can you see my mouse? Yeah?
Yep.
So we transmit light through each of these regions, and we take a look at the optical response by transmission spectra. And based on the peaks, we'll get one peak for aluminum, one peak for gold. We therefore can then take those values and put them into our mapping system. So we did that for each of the tongue devices.
Fantastic. Thank you for answering.
No problem.
Question now from Vinko. Do you want to ask it yourself, or do you want it read?
Hey, really great talk and interesting. Out of curiosity, is it possible to use this technology as a diagnostic tool for diseases? So if you're testing complex mixtures, I imagine something like spit or blood or serum would be a complex mixture as well. Would this work for some diseases?
Great question. So the answer is actually a little more complicated than a simple yes or no. The thing related to testing for diseases when you're looking at biological samples is that the way that this mechanism works is it needs to be trained. It needs to see all of the different possible capabilities and different combinations of results to be able to accurately understand what it's sensing. So in the same example I used for chocolate, if you had never had chocolate before, I blindfolded you, handed you something and you ate it, would you be able to identify it as chocolate? Maybe, maybe not. So it depends on what exactly you're running through the system. So if you're running spit, if somebody ate a hamburger before, and I've never trained my device to test saliva that has burger grease in it, then my system is not going to be able to identify accurately. So these sorts of devices can work for that, but because it's not looking for a direct analyte per se, it's not looking for one specific component in a mixture. It's looking for understanding based on the complexity of all the components, does this classify with some accuracy to a known group? So for disease recognition, typically what you want to use is some sort of mechanism in which you can find a particular antibody or a particular virus or a particular bacteria, et cetera. And so for those types of devices, it would be more based on a sensor with high specificity to one specific analyte. So you can use Plasmonics to do that, but designing a tongue device for that is not necessarily going to be the easiest method.
Zachary's got a question. Zachary, happy to read it out yourself?
Yeah, hi. I was just going to ask, did you find that the more expensive the alcohol was, the closer the data points were together in that cluster that you showed towards the end of the graph?
So the graph that I showed here, I really wish the please see the people in the waiting room would go away because it's really blocking my mess. There we go. The graph that I'm looking at here that I'm showing, the clustering is actually not based on price. It's based on variations in my device and my sensing. So the price of a whiskey doesn't necessarily have anything to do with the tightness of these groups. It's a statistical analysis technique based on multiple repetitions of reading my device. So it's basically not so much an analysis of does the complex mixture change over time. It's more of an analysis of how good is my device at being consistent with reading it. Because in theory, if I'm testing the alcohol and I'm keeping it closed and it's not long periods of time between, it's not evaporating the alcohol out, etc., my measurement should be relatively the same every single time that I measure. Does that make sense?
Mm-hmm. Yeah, thanks.
Yeah, no problem.
I had missed a question from Mata.
Yeah, I was going to ask if your device can actually be used to detect the alcohol, the kind of alcohol that a person drank by testing their breath.
Good question. Very similar to the question asked about testing for disease. You would have to get lots and lots and lots and lots of samples to test. So you could design a system in which it will build a statistical profile, and then based on someone's exhaling a breath, be able to see if it knows that and identify it. But that would take a lot of data. So yes, you can definitely do that. You would just need to, again, build a giant data set to identify and classify that.